Hi, and welcome to my OCR A-level biology revision session with me, Christine. So today's lesson, we're going to look at stem cells and cell differentiation. So to understand what a stem cell is an undifferentiated cell. It's a cell that has the ability to divide by mitosis and then differentiate. So we know that our cells have got their cell cycle and they go through the interphase, then mitotic division, then cytokinesis to produce two genetically identical daughter cells. So a stem cell is an undifferentiated cell that has that ability to divide and produce two genetically identical daughter cells, which can then differentiate. So what we have to understand is that our stem cells have got a different potency. If we start with our totipotent stem cells then, if a stem cell is totipotent, this is normally an embryonic stem cell, what we mean is that that stem cell can differentiate into any cell type. And that therefore means that it can actually produce a whole organism. So a totipotent stem cell can produce a whole organism. So when we have a zygote, a fertilized egg cell, it will go through mitotic division. So one fertilized egg cell, the zygote, then becomes two cells. Those two cells become four cells. Those four cells become 8 cells. Those 8 cells then become 16 cells. So in those embryonic stem cells, up to about the 16 cell stage, all of those cells are still totipotent stem cells. That means that they can produce a whole organism and any cell type. After the 16 cell stage, we then move on, and the embryonic stem cells are now classed as pluripotent. So they are pluripotent because they've started to differentiate. They cannot produce a whole organism anymore. However, they can produce all tissue types. So after the 16 cell stage, the stem cells are now classed as pluripotent. That's because by day 7, if we were talking about a fertilized egg cell in a human, that starts to differentiate into the blastocyst, the bit that will become the fetus, and the bit that will become the placenta. So it started to differentiate, but not quite fully differentiated. So therefore we say that it's pluripotent because it can become all tissue types, but not the whole organism. If it's the whole organism, then it's totipotent. So as we start to move through the development into the point where the fetus is ready to be birthed, once we have been birthed, what will then happen is we class our stem cells as being adult stem cells. Now, they are now multipotent, so they can give you a range of cells within a certain tissue. For example, the hematopoietic stem cells in your bone marrow give rise to various blood cells, but they cannot give rise to any type of tissue or any type of cell. So it's important that you note when you're talking about stem cells, are they embryonic stem cells up to the 16 cell stage? If they are, it's totipotent, and that gives you that whole organism. Is it pluripotent, or are we talking about adult stem cells and therefore they have to be multipotent? So pluripotent stem cells are still embryonic. They're just the 32 cell stage up to the point in which the organism has been birthed. Now, it is a case of that we can actually revert some cells back to being the previous potency, but only with specific procedures being done. And I will do a whole video on that and the uses of stem cells at another point. Okay, so if we understand our stem cells then can be undifferentiated cells, we need to then understand what it means to become differentiated cells. So we know that our cells have the cell cycle and they go through interphase. Well, you can see in my diagram, there's actually a section here which is called G0. Now, this is when a cell is actually going to leave the cell cycle. So if a cell leaves the cell cycle, it is no longer able to go through mitotic division. So when a cell differentiates, when those genes have been switched off, that then means that that cell no longer has the capacity to go back into the cell cycle and therefore no longer will go through mitotic division. And that's because as cells differentiate, they change potentially their shape, that it may be non-symmetrical and you end up with this asymmetry, which results in you not having your two poles for the centrioles to move to. So therefore, if a cell differentiates, it will leave the cell cycle. If there is any DNA damage, then we don't want DNA replication to occur with that damaged DNA. So because of that, what happens is the cell becomes senescent, and that means it moves into the resting phase, G0, and that is where it's no longer able to divide, but still metabolically active. So even though the DNA is damaged, it's still able to do a lot of those chemical reactions that are needed. We just don't want to replicate the DNA because there has been some form of damage that has occurred, and that damage would not be able to be repaired. Also, aging. As we get older, there is an increase in the senescence of these cells, and therefore that links with many age-related diseases. So as you get older, your cells then tend to have their DNA damaged more. They become senescent where they're not able to divide, but they're still metabolically active. But because they can't divide, they can't replace and repair the tissue that has been damaged. And that's why we end up with those age-related diseases. Now, it is important to note that when cells differentiate, when they leave the cell cycle and they go into this resting phase, some of them can actually be stimulated to re-enter that cell cycle. So although differentiation means that they leave the cell cycle, with some they can re-enter and start dividing again. So for example, lymphocytes are an example of a cell which is going to be re-entering the cell cycle if we need to do clonal expansion. And you'll learn about that in the communicable diseases video, so do please check that one out. Okay, so the types of differentiated cells that you have to know about are very specific. So erythrocytes then. Erythrocytes are your red blood cells, and I have covered this in my video on the blood vessels, but just to cover it again here, our erythrocytes are biconcave in shape, and that biconcave shape is going to make there be a larger surface area to volume ratio. They have no nucleus. They have no organelles. So what happened is, as the cell was differentiating, it then removed the unnecessary organelles, and therefore it allowed for more space for containing that hemoglobin. Now, hemoglobin is a globular protein that was made through protein synthesis. So therefore, the DNA had to be present to make the hemoglobin, and then when it was no longer needed, then it was removed. So it's important to note that as the cells differentiate, they change to have that specific function. In this case, the hemoglobin allows for that transport of oxygen and carbon dioxide. Now, another thing about the erythrocytes is they're very flexible in their shape. That's because they need to squeeze through those capillaries. Those capillaries have a very small diameter, only slightly greater than the erythrocyte's diameter. And that therefore means that as those erythrocytes squeeze through the capillary, they are very close to the capillary, and that's going to shorten that diffusion distance and increase the surface area that's being exposed. Now, your neutrophils are another cell you need to know as an example of a differentiated cell. So a neutrophil is a type of white blood cell. We can tell a neutrophil is present by its multi-lobed nucleus because it is going to be squeezing around, and because it makes lots of proteins, it has this multi-lobed nucleus. Now, the granular cytoplasm in a neutrophil is identifiable because that is the place where the lysosomes are going to be present. Now, those lysosomes contain the lysozymes, and that's important to remember that your neutrophils are a type of phagocyte. So again, I have covered this in the communicable diseases topic, but please do go check those videos out if you haven't already. So it's important for us to note that our erythrocytes and our neutrophils are differentiated cells, and remember they actually came from the hematopoietic stem cells which are found in your bone marrow. Now, it's interesting to know that erythrocytes only have 120 days in which they actually can be transported around the body before they are no longer useful. So therefore they have to be replaced. Now, because they have no nucleus, they can't go through mitotic division, they cannot make more of themselves, so therefore they are replaced by the hematopoietic stem cells which are found in your bone marrow. Well, your neutrophils actually have a much shorter lifespan. They only last about 6 hours. And so therefore, these hematopoietic stem cells have to be able to go through mitotic division and then that differentiation process to produce the erythrocytes and the neutrophils. So the hematopoietic stem cells are multipotent because they are going to produce the blood cells. So they've already started to differentiate. They haven't fully differentiated yet because they haven't differentiated into an erythrocyte or a neutrophil, but they have differentiated into a blood cell. Now, a few other examples you need to know are the squamous epithelial cells. So I did cover these before, but again, just to go back, so this links to your exchange surfaces. Your squamous epithelial cells are flattened. They are squashed epithelial cells, and epithelial cells cover things. So your squashed epithelial cells are flat in shape. That makes them very thin, one cell thick, short diffusion distance. It means they can fit together. They can form this really tightly fit pavement, and that therefore means that only when there's high enough pressure can things be pushed through the gaps between the cells, the fenestrations. So when you're looking at the tissue fluid formation video, that comes into play. And when we're looking at the exchange surfaces, when we talk about the diffusion of the oxygen and carbon dioxide, it's that very thin, one cell thick, short diffusion distance. Now, another one we have covered again in exchange surfaces is your ciliated epithelium, which is made up of your ciliated epithelial cells. So your ciliated epithelial cells are your differentiated cells. Now, these are differentiated because they have these cilia to beat in rhythm. They're going to move that mucus, those trapped pathogens. But you also have ciliated epithelial cells in your oviduct if you are female, and that therefore allows for this beating in rhythm and moving the egg to the uterus in preparation for that fertilization. So ciliated epithelial cells are important for this movement of the substances, whether that be the mucus and trapped pathogens or whether that be the ovum. Now, another one you have to know is your sperm cell. So the sperm cell is the male gamete, and it has some very key features which is important for it to carry out its role. So the first thing that we talk about is the flagellum. Now, the flagellum is made up of protein to move the sperm cell towards the ovum, that egg cell. So therefore, we need to remember our cytoskeleton. Our cytoskeleton is made up of our microfilaments, and they are therefore going to allow for this movement. And that also links to the structure, which is the 9+2 structure of the cytoskeleton, because we're looking at a eukaryotic organism. Another thing to note is there's many mitochondria. If there's many mitochondria in the mid region of the sperm cell, that's going to mean that aerobic respiration can occur. Aerobic respiration you will learn about in module 5 in more detail, but basically that's the energy needed to move the cytoskeleton. So it will supply the ATP molecules, and those ATP molecules will be used for that movement. Also, because we're talking about a gamete, this links to your meiosis topic here, in the fact that the haploid nucleus, meiosis has occurred, we now have a haploid nucleus, and that is to ensure that the zygote, that fertilized egg cell, when that sperm cell and egg cell fuse together, that it contains the correct diploid number. So we need to have half the genetic information from the male gamete and half the genetic information from the female gamete, so that when they come together, we end up with a full set of chromosomes, our diploid number. Now, as I said before, the male and the female gametes need to fuse. Well, the female gamete actually has a protective layer. So the male gamete has to differentiate to have a specific region, which is called the acrosome, which contains digestive enzymes. And those digestive enzymes actually digest that protective layer of the ovum to allow the sperm cell to get in and fertilize the ovum. Now, it is important to note that we only ever want one sperm cell nuclei to fuse with the female nuclei, and therefore, because of that, by having this process of that digestive enzymes needed to break that protective layer down, it means that only the sperm cell that's able to do that in the quickest amount of time is going to be the most successful at fertilizing that egg cell. So it's an advantage that the sperm cell has this acrosome. It's a necessary point to ensure that there is only the correct number of diploid chromosomes in the fertilized egg cell, the zygote, and not polyploidy where there would be more than that. Okay, so let's move on to plants then and have a look at some of those differentiated cells. So the ones, again, that you need to know are your root hair cells. So your root hair cells have got these long extensions. They're what we call the root hairs. So those long extensions are going to increase that surface area. The root hair cell needs to absorb water and mineral ions from the soil so that it can be transported up through the plant system. So having these long extensions, these root hairs, means that it's going to increase that surface area. But as I said before, it needs to absorb and take in these mineral ions and water. So therefore, we have to think about our membranes. We have our cell wall, but we also have a membrane which is selectively permeable. So therefore, it's differentiated to have very specific channels to allow the molecules that it needs into its cell and into the plant to get across. So not only does it need to have the membrane channels, it also needs to have the mitochondria. So in the root hair cells, having many mitochondria, again, aerobic respiration, that is going to provide the ATP molecules, those that are necessary for active transport. You will look at this in more detail if you check out my transpiration video, but just so you are aware that ATP is needed for active transport. Mineral ions get pumped against the concentration gradient through the ion channels, and that is going to affect the water potential, meaning water will move in by osmosis. So if we move up the plant then and we look at the Palisade cell. So the Palisade cell is a very specific cell. It is found in the leaf of the cell, and it has a very large vacuole. Now, the large vacuole in the Palisade cell means that it can maintain turgor pressure. Now, the Palisade cells are at the top region of the leaf, and they are known as the Palisade mesophyll when we look at it as a tissue. So as you can see in my diagram, they are really long and they are really packed together, and that makes sure that they are maximizing that absorption of light that's going to come from the sun. Not only are they packed together and really long columnar shaped, they also have lots of chloroplasts. Now the chloroplast, you'll learn much more on this in module 5 when you do photosynthesis, but the Palisade mesophyll cells contain lots of these chloroplasts, so that maximizes the absorption of light for photosynthesis. So this cell has differentiated to have a very specific shape and structure for the role that it plays within the cell, within the organ. Now, another one you have to know about is your guard cells. So your guard cells are again found in the leaf. Now, these are necessary for gas exchange. When a plant does photosynthesis, it needs carbon dioxide. To get the carbon dioxide in, it needs to have these openings. So they are found on the surface of the leaves, normally at the bottom, but they can also be found at the top, depending on the plant. And they are called stomata. So the guard cells are either going to open the stomata, these openings, or they are going to close the stomata. And they do that because they have very thick walls on the inside compared to the outside of the cell. So the wall on the inside is much thicker. And it's also to do with the movement of substances. So going back to your biological membrane, thinking about the transport across the membrane, it's important that you note that if we want to open the stomata, the potassium ions are pumped in to the guard cells. Now, if they're being pumped into the guard cell, that use of the word pumped tells you it's active transport against the concentration gradient. So if we're going against the concentration gradient, we are therefore creating a higher solute concentration inside the cell. Therefore, water is going to move in, down its water potential gradient, and as the water moves in, those cells, those guard cells, become very turgid. And when they become turgid, because of that thicker wall on the inside, it causes this curvature of the guard cell, and that is what opens the stomata. So if we want to close the stomata, if, for example, there is drought, what will happen is it will be the opposite. The potassium ions are pumped out, the water will leave, and therefore it becomes more flaccid. The cells are more flaccid and they will close together. So this is important that you understand that these cells are differentiated in a way that allows them to carry out their function. So when we know that a cell, for example, a Palisade cell has differentiated, and then we have a group of these differentiated cells working together, and they have a very specialized function, we call that a tissue. So there are quite a few tissues that you need to know about. The squamous epithelium, which we talked about and you will talk about in exchange systems again. The ciliated epithelium found in the trachea, found in the oviducts. The cartilage, which is found in your trachea. Again, going back to other videos that will help you with this knowledge. In module 5, you will learn about muscles and the different groups of differentiated cells that are necessary for muscles to contract. And when you check out my transpiration videos, you'll learn more about your xylem and your phloem tissue. Now, these groups of differentiated cells, these tissues, then can work together to produce an organ. So the leaf is the organ. The organ is made up of all these different types of tissue. So we have the waxy cuticle, we have the upper epidermis, we have the Palisade mesophyll, we have the spongy mesophyll, we have the lower epidermis, which has the guard cells, and then we have a waxy cuticle on the bottom as well. We also have in between what's known as the vascular bundle, which contains the xylem tissue and the phloem tissue. So it's important that you note that cells will group together to become tissue. The tissues that group together become the organs, and the organs that are coordinated together can become a system. Our transport system, for example, when we talk about the movement of water through the plant from the root to the xylem to the leaf, that is through the transport system. When we talk in humans and we're talking about our blood cells, we're talking about the blood vessels that were made up of cells that became tissues that then became the organ, the vessel, that then became the organ system, your circulatory system. So if you like this video, then please do click on the like button and subscribe to my channel. Also, do check out my revision platform, www.aiqchat.com, which should help you with your revision.